Hello, everyone, and welcome to our last lifeline. This week, we will be looking at the genetic code. This is week 15, and as usual, you will be getting some PPP points if you do the PPP quiz. A couple of reminders. Be sure to check your Canvas grades. Whenever you see this presentation available, it will come along with an announcement letting you know that all your grades are up to date at this point and that you should have a look at them to be sure that there is no mistakes. In addition to that, we have a couple of assignments left. That includes your week 15 PPP quiz and the spot surveys that are due on April 24th. Last week I told you that we were not sure if we were going to give points for this yet. Now we are. You will be getting 1 point if you do this spot survey. All you have to do is go to the spot website, complete the survey for Lifeline under my name, and then just do your regular and real feedback because it really helps me. Then all you have to do is submit proof of the completion. Still, I don't need to see your answers or anything. I just need to see that blue square that says my name, Alex Rodriguez, and that it says survey completed at the bottom. If you're able to upload that to Canvas, then you will be getting that 1 point. So let's get into it. What is DNA? We are going into the realm of molecular genetics at this point. We're looking at how genetics work down to the molecular level. This is all we will be doing today. So what is DNA in a molecular sense? First, it is a carrier of genetic information. It is made of two long antiparallel and complementary strands that are paired together. Antiparallel just means that they are going in opposite directions. Now, this also implies that each strand must have a direction. That direction can be either from 5' to 3', and this is what we're looking at on this picture right here. They are antiparallel because, as you can see, this strand is going from the 5' end to the 3' end, whereas the opposite strand is going from the 3' end to the 5' end. That is antiparallel. Complementary refers to what's going on here with the bases. For example, A complements with T and C complements with G. And that is what you see all across the DNA strand. The two strands connect to form a double helix. Now, they don't look as straight as they do here. This is just for clarity. And they also don't look like what you see here. Alright, if anything, they look like a combination of both of these, because both the nitrogenous bases and the backbone are made up of carbon skeletons, alright? There is a lot of carbon and nitrogen and phosphorus. There are several elements that go into making these. Fortunately, you do not need to know these for this class. But just be aware that it is by far not as simple as it looks here. Each strand is composed of many individual nucleotide monomers. A monomer is only a single piece that makes the DNA strand. If you call it a polymer, then you have many monomers, if you will. That is all that means. And this serves as the blueprint for protein synthesis. Now, this takes us back to the central dogma, which we've been talking for a while now, right? The central dogma, again, states that DNA transcribes to RNA, and then RNA translates to proteins. That is the central dogma. That is how this is the blueprint, because DNA does not directly synthesize proteins. It has to make RNA in between, and then it is the RNA that is able to synthesize the proteins with the help of the ribosome. Nucleotide structure. At first, there is a nitrogenous base. It can be a purine or a pyrimidine. You do not need to know the differences between each of these. You need to know that there are 4 kinds that exist. Actually 5 if we count RNA. but since we're talking about DNA, let's call it 4. If anything, what I would want you to know is the difference between purines and pyrimidines. The great difference, and please know that a purine is not all of this. It is not all of this that you're looking at here. This is the structure of a nucleotide, not a nitrogenous base. A nucleotide is composed of 3 things: a phosphate group, a carbohydrate, and then the nitrogenous base. In this case, this nitrogenous base, we are looking at a purine. Purine are 2 ring structures. Pyrimidines are single ring structures. So that is the nitrogenous base. Then the pentose sugar, or the carbohydrate, is the other part of the nucleotide. And then there is the phosphate group, right here. 3 parts to a nucleotide. Now let's look at how these nucleotides together make up the DNA structure. The nucleotide, which is composed of the nitrogenous base, the pentose sugar, and the phosphate group, is the basic unit of DNA. Then there are the 4 important nitrogenous bases. These include adenine, guanine, cytosine, and thymine. It is important that you know the names, okay, and that you know that they exist. And honestly, that is pretty much all you need to know for this class. Bases on each strand paired together via hydrogen bonds. And what I mean by this is all you need to know about these is that you do not need to know the structures, okay, or tell them apart by looking at the structure. No. If anything, you just have to be able to tell if it's a pyrimidine or a purine. For example, purines include adenine and guanine, cytosine, thymine, and the 5th one, uracil, which is only found in RNA, and instead of thymine, all of those 3 are going to be pyrimidines. As I mentioned before, purines are a 2-ring structure, and then pyrimidines are a 1-ring structure. Adenine and guanine are purines, and then the cytosine and thymine and uracil also, which is not here, but uracil does exist. It is found in RNA, and instead of thymine, those are called pyrimidines. Now, this is the bonding that occurs between purines and pyrimidines. First of all, this is the rule. You have to pair a purine with a pyrimidine all the time. That is how they pair. Now, there are 2 types of pairing that can happen in DNA. You can pair cytosine, which is the pyrimidine of the pair, with a guanine, which is the purine of the pair. This pair of cytosine with guanine makes 3 hydrogen bonds. Now, the other pairing that's possible is adenine, that's the purine, and thymine, that's the pyrimidine, and this one makes only 2 hydrogen bonds. These hydrogen bonds are what provide the stability to the DNA double helix. And these other have the 2 bonds, as I mentioned. Now, let's dive a little bit deeper into the pentose sugar, which is the other part of the DNA structure. First, it is going to be a pentose sugar. Pentose implies that there's 5 carbons in the structure. And these carbons are numbered from 1 through 5. That is how we get the 5' and 3'. Let's look at it. We begin counting from this carbon. It's usually referred to as the anomeric carbon. But this is where we begin, alright? This is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. Now, before I even show it to you, I want you to realize how the directionality is going to be seen. From 5' to 3', okay? This is what we keep saying for DNA over and over again. The directionality is from 5' to 3'. Now, let's look at the difference between deoxyribose and ribose. Deoxyribose is missing an oxygen. That is why it's called deoxyribose. Deoxyribose is missing an oxygen in comparison to ribose. Now, let's have another difference looked at. Deoxyribose is going to be found in DNA. How do we know it's going to be found in DNA? If you know what DNA means, it means deoxyribonucleic acid. So, deoxyribonucleic acid, alright? Now, ribose is going to be found in RNA, and in a similar way, RNA means ribonucleic acid. So just ribo, not deoxyribo, alright? In DNA, the sugar is deoxyribose, and in RNA is ribose. I just told you what the names mean. I hope this helps you. Phosphate group. Now this one is the simple one of the three components that we've already looked at. But it's still pretty important because this is the one that's responsible for making the backbone, for making the links in the backbone. This is what actually holds the backbone together. The hydrogen bonds that we discussed earlier are what keep the two strands together. Now, these phosphate groups are what keeps the strand itself together. Now, these phosphate groups are bound to the 5' carbon on sugar. As you can see here, this is the 5' carbon, and this is the one that's bonded to the phosphate. Here is another 5' carbon. It's bonded to the phosphate. This forms the sugar phosphate backbone. Covalent bonds connect phosphate to sugar and forms long chains of nucleotides. Very long, by the way. Like, long does not begin to describe it. There are millions of these units in a DNA molecule. Notice that the 3' and 5' ends and how nucleotides always are added to the 3' end. Remember, a nucleotide is the collection of the three things: a phosphate, a pentose sugar, and the nitrogenous base. This is what we're looking at. Here, we're looking at how a new nucleotide is being added to an already existing strand of DNA. As you can see here, the phosphate connected to the 5' end of this nucleotide is being added to the 3' end in the already existing DNA molecule. And this is why DNA is said to be made from the 5' to the 3' direction. That is how DNA synthesis occurs. It will always be the 5' end being added to the 3' end, which in the end just leaves a 5' end open and a 3' end open, as you can see here. Now, this picture may look a little bit complicated, granted, but if you're able to define what we're looking at, you will certainly understand what we mean by the directionality. Again, if you understood how to count the carbons, you will see that this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So this is the last carbon that we're able to see here, and hence is the 5' end. If we go all the way down, let's look at the carbon that's empty. 1, 2, 3'. This is the empty carbon. It ends in the OH. So that is the directionality, 5' to 3'. Because now you understand that DNA has an anti-parallel directionality, you know that the other strand has to be going in the opposite direction, from 3' to 5'. Let's talk about Chargaff's rule. This rule is basically the ratio that DNA has to follow regarding the nitrogenous bases. This rule states that DNA from any cell would have a 1-to-1 ratio of pyrimidine and purine bases. But it gets a little bit more specific than that, as follows. The amount of adenine is equal to the amount of thymine. Alright? So adenine has to be equal to thymine. And if you think about it for just a minute, you will understand why this has to be the case. Because A only pairs with T, and G only pairs with C. And yes, this is forgetting about mutations for a second. But in ideal conditions, and most of the time, it really is how this happens. A pairs with T, and G pairs with C. And if this is the only true real connection that can be made, then the amount of T must be equal to the amount of A at all times, and the amount of G must be equal to the amount of C at all times. In humans, there are four bases. The four bases are present as follows. Adenine is present by 30%, and the thymine then must be present by 30% as well. Yes, there is a little bit of discrepancy here, but this only has to do with the accuracy of the counting mechanism that the scientists were using. Or, yeah, some mutations that are possible to happen. But in its essence, you see how similar the numbers are. And then guanine, 19.9%, and cytosine, 19.8%. So basically 20% and 20%, guanine and cytosine. 30% and 30%, thymine and adenine. And if you add all of these together, you get 100%. Now let's see if you understood the rule. So if you have 40% of adenine in your DNA, how much thymine, guanine, and cytosine is present? And if you understand Chargaff's rule, you will see that this is really all you need to know in order to answer the question. So let's give it a shot. So we have 40% of A, right? A equals 40. Now, what does A pair with? We already know that A pairs with T. And according to Chargaff's rule, A equals T. So T must be 40 as well. Now, how are we going to get the amount of C and G? Well, let's see how much 40 and 40 gives us together. 40% and 40% gives us 80%. And of course, we have to have 100% of our DNA, don't we? So how much does that leave us with? That leaves us with a 20% difference, all right? 20%. This is what we're working with for C and G. Now, because C and G must be equal to each other, how can we split 20% to have equal numbers? Easily, we arrive to the conclusion that C must be equal to 10%, and therefore G must be equal to 10% as well. And all of this would give us the total of 100%. There you have it. That's Chargaff's rule for you. DNA replication. Why do cells need to replicate their DNA? So that cell division can occur correctly. Mitosis and meiosis. This is absolutely important. It is imperative that the DNA replication process happens with an extreme level of accuracy. Because if this process is not accurate, we would not really be able to engage in mitosis. Remember, mitosis produces clones of itself. And these clones are important, especially in processes in our body, such as, think about when a patient undergoes a liver transplant, okay? That liver has to regrow after it's transplanted. And all this process happens through mitosis. This is how important mitosis is. And in order to do correct mitosis, you need to do correct DNA replication, because mutations are bound to be troublesome in the long run. This allows organisms to grow tissues and sexually reproduce. Now, the example of liver has to do more with mitosis. Now let's talk about meiosis. If you understand meiosis, you know that these cells have to replicate their DNA once only once. Because even though there are two cellular divisions during meiosis, replication only occurs before the first division. And during the second division, it does not occur again, which is how we end with 4 haploid cells. Alright? That is why DNA replication is important. It is also important to create the protein, absolutely, because that is what we need. We need the DNA that is going to code for RNA, so DNA makes RNA, RNA makes proteins, so DNA in the end is actually making proteins. That is why we say the DNA is a blueprint for protein formation. Because without DNA, there is no way we could get mRNA that is going to code for those proteins. DNA replication occurs in a semi-conservative mechanism. This means that two DNA molecules are produced in the end, and that the strands act as a template to synthesize a complementary strand. So semi-conservative means that a part of the DNA is conserved, and a new part comes along. As you can see in this example right here, we have our original DNA molecule, and we show the semi-conservative process by looking at the second generation of DNA. Now we see we have an old strand and a new strand, right? And then in a new division, we actually only have two old original strands out of the 8 strands that are available now. This is showing semi-conservative replication. Each DNA molecule contains an original strand from the template and one new strand. Now, here we're comparing it back to the original parent strand. But if you think about this one is going to be divided, it's going to be separated into 2 strands, and then each strand is going to serve to make 2 new strands. That is really the definition of semi-conservative mechanism. Now, there are variations in replication. As we know in science, there's always a variation of the process, right? So first, let's look at the differences between prokaryotes and eukaryotes. In prokaryotes, there is only one origin of replication. An origin of replication just means where replication began in the first place. Prokaryotes are known to have one single chromosome. They have one single circular chromosome. It is a loop of DNA, as you can see here. And because it is a loop, only one origin of replication is seen. And really only one origin of replication is needed, because their genome is not as large as the genome of a eukaryote, for example. But that aside, you can see how with only one origin of replication you would be able to get 2 new DNA molecules. Now, if we look at eukaryotes, we will see that there are multiple origins of replication. And think about it. Eukaryotes, let's talk about humans. We have 46 chromosomes, right? When replication occurs, we end up with 46 duplicated chromosomes. But what is different about our chromosomes in comparison to the DNA of a prokaryote? Besides the fact that we have more chromosomes, our chromosomes are linear. They are linear. They are not circular. So, multiple origins of replication can be used just to make the process go faster. Eukaryotic DNA is longer and it takes more time to replicate. Multiple origins help speed up the replication time. And then the mechanism of DNA replication. Now, let me give you a full disclaimer here. The mechanism of DNA replication that we're about to get into is far more complicated than what I'm about to tell you. This is good news for you, really, because being totally honest with you, I did not completely understand DNA replication until I took genetics. So if you do not completely get it at this point, please do not be afraid to do bad in the exam, because you're really not expected to be able to recite the DNA replication process. Not until you take genetics at least, or when you take biochemistry. So right now, what we are aiming for is that you realize that there is a mechanism, and that this mechanism works in a certain way, which we are about to explain. But please do not be flustered if it turns out to be a little bit too much, alright? So, the initial DNA molecule has two complementary strands of DNA. This we've already set in stone. This we already know. The first step of a replication is to separate or unwind the complementary DNA strands like a zipper. Alright? That's the first thing. We have to take them apart. If we're going to replicate DNA, we have got to take it apart in the first place. Each conserved strand now serves as a template for the addition of nucleotides along the new complementary strand. Nucleotides are connected to form the sugar phosphate backbones of the new strand, and as we have already well established, this synthesis of the new strand is going to occur in what direction? If you said 5' to 3', you are correct. According to the semi-conservative model, each newly produced DNA molecule now consists of one conserved strand and one new strand of DNA. So here you have it. This is going to be basically everything that we're about to get into. We're going to get into the individual components one by one. So let's have a look at it. First, we're going to have a look at helicase. Helicase is going to be the molecule responsible for unwinding the strands, for taking them apart as a zipper, as we mentioned before. There are other things here that we are not going to get into, alright? For example, this molecule here, I'll just mention it, but we do not have a definition for it. This molecule here is called gyrase. Gyrase is responsible for really untwisting the DNA. As you can see, DNA everything is going in this direction, okay? From here to here, from left to right. Before gyrase gets to it, the DNA is still wound. Whereas behind where the gyrase has already happened, it is no longer wound, and helicase is able to separate the two strands now. Then everything else is happening. DNA primase comes in here, it leaves a primer. We will see what a primer is, don't worry. Then we have DNA polymerase. This is the one that actually does the work of synthesizing DNA. What else do we have? We have DNA ligase. This is going to be responsible for, as the name implies, ligating the fragments that are left behind. Let's get into a little bit more detail. And the first thing I want you to notice is that there is such a thing called as a leading strand, and there is also a lagging strand, and we will see the differences. Hold on. So the first step, as we said before, is going to be to unwind the DNA. And this is going to happen by the enzyme helicase. This is the enzyme that unwinds the parental double helix at the Y-shaped replication fork, near the origin of replication. What do we mean by the Y-shaped replication fork? It's this portion right here. This is the Y-shaped. If you look at it, it looks like a Y, yeah? This is the replication fork. It breaks the hydrogen bonds between bases. That is how helicase works. Remember, what is keeping the DNA strands together are those hydrogen bonds. If this enzyme is coming along and is getting rid of the hydrogen bonds, then there is nothing else holding the strands together. If you think of DNA as a zipper, helicase unzips the zipper. There you have it. Then we have these small proteins here. These are called single-strand binding proteins. These are really important because just know that the work that helicase is doing is not permanent. It may be able to break apart the hydrogen bonds for a time. But then, if there is nothing keeping these strands from getting back together, then there is nothing keeping them from reforming the hydrogen bonds. This would be called reannealing in molecular genetics, alright? So basically, there is nothing preventing the zipper from closing again. unless we have these single-strand binding proteins, which literally are going to keep the strands apart from each other. So they stabilize the unwound parental DNA and holds the two strands apart, forming a replication bubble. A replication bubble is basically the whole space that is being formed by the helicase and by all the other enzymes that are working at the same time to make new DNA. This is basically what we're looking at. This is the parental strand, as you can see here. Actually, the parental DNA. We have the two strands. Replication fork being formed by helicase, and replication bubble is everything that's being left behind. Yeah, let's look at it again. The original DNA molecule, helicase is coming along, making the replication fork, and leaving the replication bubble behind. Here we can see that gyrase already passed along and already unwound the DNA. Now, DNA replication, the actual synthesis of DNA, begins with RNA primase. RNA primase lays down RNA nucleotides to make a primer. A primer is this portion right here. A primer is necessary in order for DNA to begin being produced. Now, why is a primer necessary, you may ask? This is because DNA polymerase, which you can see up here, DNA polymerase is only able to begin synthesizing DNA if it can get a hold of an empty 3' end. We're going back to the 3 and 5' ends, okay? Remember that nucleotides are always added to the 3' end, from the 5' end to the 3' direction. So a 3' hydroxy, a 3' end is needed in order to begin adding new nucleotides in DNA. That is what the primer is doing. The primer, because it's RNA, does not need anything to begin synthesizing. Here in this portion you can see how DNA in red already was laid out after the primer. We do not have DNA here yet. We can guess that DNA is going to be formed from here to here in a little bit, right? But notice how DNA primase was able to just put its RNA nucleotides on the strand without there being anything else here to begin with. So, as I was saying, this provides the 3' hydroxy end for DNA synthesis to occur. That is the work of DNA primase, because DNA polymerase needs that 3' hydroxy in order to lay down its nucleotides. Primase moves toward the replication fork on leading strand and away from the fork on the lagging strand. We will see the movement of this in the next video. Here, that is the primase. There you have it. And then this leaves the 3' hydroxy here. Now, DNA polymerase is ready to just come along and begin laying its polymers, its monomers. Now, DNA polymerase, that is the one that actually does the work of synthesizing new DNA. It adds the new DNA nucleotides to the template strand to form the new complementary strand of DNA. Here is where we see the difference between the leading strand and the lagging strand. The names really tell you a lot of what's going on. The leading strand, the nucleotides are continuously added in the 5' to 3' direction toward the replication fork. So, here we're looking at the leading strand. The DNA is being unzipped from right to left in this example, okay? So helicase is going here, opening the replication bubble and the replication fork, making it right here, yeah? The leading strand first had a RNA primase come here. It laid a primer, as you can see there. That's the primer. Then DNA polymerase is able to come here and take advantage of that empty 3' hydroxy and begin laying its nucleotides. Now, that DNA polymerase is moving together with the direction that the helicase is going in. So, this DNA polymerase is just following helicase, and therefore it does not need to unattach at any point. It can just follow it. It keeps following, and it doesn't stop until something stops it. Alright? So that is why this is called the leading strand. It is continuous. Now, the lagging strand, on the other hand, is discontinuous. Let's understand why. Nucleotides are not added continuously in this strand. DNA template runs from 3' to 5' from the replication fork, and since DNA is made from the 5' to 3' fragments of DNA are made, known as Okizaki fragments. So let's try to show this in a better way. So, as you can see here, DNA polymerase, it's actually reading the leading strand from 3' to 5'. Look, to 5', yeah? It's reading it in that direction, but it's actually synthesizing in the 5' to 3' direction. Now, on the other hand, right here, if the DNA polymerase needs to read from the 3', look, from the 3' to 5' direction and synthesize in the 5' to 3' direction, it is going to have to do it in a discontinuous way, because helicase is unwinding the DNA towards this side, okay? Therefore, several RNA primers are going to be needed. RNA primers are going to lay a primer here so that DNA polymerase can come and synthesize from 5' to 3'. This is going to keep on winding a little bit. After a little bit, just a little bit, another RNA primer is going to be needed, and it's going to be laid somewhere here, for example. After this unwinds, then DNA polymerase is able to fill in that gap again, and it does that over and over again. This is how it's done it. The red dot is our DNA polymerase, okay? It came here, pop, it synthesized this fragment. It's done. Then it came back here to the other primer, synthesized this portion, stops. It comes back here to the other primer, synthesize, stops. Goes back to the primer that has not been added yet in this picture, synthesizes 5' to 3', stops. And it does that over and over again, over and over again. Let's look at the video. This is the leading strand. There is only one primer here. This is DNA polymerase, and it goes. It goes on and on and on and on and on, and nothing will stop it until the DNA is over, or until it meets another portion that has already been synthesized. Remember that there are several replication origins in eukaryotes. So if that happens, then it stops. But now let's look at the lagging strand. So, there was a primer there already. We can see DNA was already synthesized here. RNA primase already left a primer here. DNA polymerase can now come and synthesize here. RNA primase has to leave another primer here. And this is the same video in a loop, but really this is all that's happening at the molecular level. Look at it again. 5' to 3'. RNA primase puts another primer. Boom, there it is. DNA polymerase can do it again. 5' to 3', taking advantage of the 3' hydroxy here. RNA primase leaves the primer again. And one more time, the DNA polymerase does its work, synthesizing the new strand of DNA from 5' to 3'. Okay? And it does it over and over and over again. DNA replication, the DNA polymerase. DNA polymerase actually has more than one function. It does not have to only synthesize from 5' to 3'. It can actually fix mistakes as well. This is it can do it with something that we call exonuclease activity. With that exonuclease activity, DNA polymerase is able to remove the RNA primer and then replace it with DNA nucleotides. This is how the Okizaki fragments are filled. This is how the gap is filled. Okizaki fragments are all of these. Every one of the DNA fragments in the lagging strand is called an Okizaki fragment. So when the primer is released, it's taken away by the DNA polymerase and filled again with more DNA. That is effectively getting rid of an Okizaki fragment. DNA polymerase also has proofreading activity to check for any errors in base pairings. So if it notices that there is an error, DNA polymerase is able to go back, remove the wrong base, and replace it with the right base. This is what we're looking at. This is the exonuclease activity of DNA polymerase, removing the primer, and then it just comes back to lay the new DNA primers. However, notice this right here. It's kind of hard to see, but there is a small cleft left, all right? It is not completely solid yet, and that is what we will look in our next one. The enzyme ligase is going to be responsible for literally ligating those ends right here. Look at it. There is an open end there, and that has to be ligated, and it will be ligated with ligase. It joins or ligates the Okizaki fragments together in order to form a continuous complementary strand. That is all ligase does. It creates a covalent bond between the 5' phosphate and the 3' hydroxy of an adjacent nucleotide. It does not add any new nucleotides. It is just getting rid of the Okizaki fragment. It is a fragment until it has been ligated by ligase. So if you look at here, it pretty much does not even look like it's doing something, but in reality, it's closing in those gaps. Now, I want you to notice something. We're pretty much at the end of replication here, but ligase is present in the leading strand as it's present in the lagging strand. So if the leading strand does not have any Okizaki fragments, why do you think ligase is even going in there? Well, this is because even at the beginning of DNA replication, there was a primer laid at the beginning, wasn't there? Because it needed the 3' hydroxy. That's one of the reasons. After that primer is removed and replaced with DNA, then that has to be ligated. Then again, we already said that there are multiple origins of replication in eukaryotes. So if this is a eukaryotic DNA molecule, when two replication bubbles come together, there may be a small cleft, as we mentioned before. Pretty much like a huge Okizaki fragment, if you want to think about it that way. And then that one part has to be ligated. And ligase would do that on both the leading strand and the lagging strand. But if you were asked, for example, which strand has more ligase function, that would be the lagging strand, because Okizaki fragments are present all along the way. So if you have any questions about this fairly complicated process, please reach out to me. You can do it through the discussion board, or you can shoot me an email, and I will be sure to answer it for you. It has been a pleasure to be your lifelong leader, and please, I will be expecting those spots. Any feedback that I can get, I will really appreciate. Thank you so very much. and I hope you all had a good semester despite the circumstances. Goodbye.